Hello, my name is Skylar Kosegi. Today is January 16th, 2024, and my presentation is on the regulation of sperm maturation. So spermatogenesis is developing spermatids, and this occurs in the testes, or more specifically in the seminiferous tubules. And you can see to the pictures to the right that the original germ cell is called spermatogonia, and they develop in between those Sertoli cells, and eventually when it reaches the lumen of the seminiferous tubules, the spermatid should be developed through different phases. So first does mitosis, so that way there's still germ cells available, and then 2 rounds of meiosis, which makes 4 haploid spermatids per spermatogonia. And then these spermatids will travel to the epididymis, where they will be developed even further. But the important thing about the anatomy behind this is that these Sertoli cells have receptors for testosterone and FSH, which both play an important role in the regulation of this process. So then here's a diagram showing the steps of spermatogenesis, going to primary spermatocyte to secondary spermatocyte through meiosis, and then meiosis again for spermatid. So again, this is regulated through hormonal controls. So the first step, or the first thing that has an impact on this process, is GnRH. This hormone's secreted from the hypothalamus, and it signals the secretion of FSH, or follicle stimulating hormone, and LH, luteinizing hormone, which are both secreted from the anterior pituitary, and then these will travel through the bloodstream to reach different parts of the testes. So LH is acting on Leydig cells, and it is signaling to them to produce testosterone, and then this testosterone will then act on Sertoli cells, and it increases, or it promotes, the maturation of spermatozoa. And then FSH travels through the bloodstream and binds directly to the Sertoli cells, and it also increases spermatogenesis in a similar way to testosterone. However, there's also negative feedback controls in this process. So you can see here when FSH levels are high and Sertoli cells are increasing spermatogenesis, they're also producing inhibin, which is a hormone, and that's traveling back to the brain, specifically the anterior pituitary, and it is inhibiting the secretion of FSH. So lower levels of FSH will decrease the amount of spermatogenesis as a negative control of the amount of sperm that's made. And then testosterone, which again is made from those Leydig cells, it's increasing spermatogenesis when it acts on Sertoli cells, but when its levels are high, it will reach back to the brain through the bloodstream, and then it's inhibiting GnRH in the hypothalamus, and it's actually also inhibiting the hormone secreted from the anterior pituitary to decrease the amount of sperm that's being made because it's not as needed. And then another way that sperm is being regulated is to have systems in place or mechanisms in place to eliminate sperm that may have problems. So sometimes sperm are damaged, they may be dysfunctional, like have motility problems or other things, or there might just be an excess amount of sperm. So they can undergo apoptosis, which means programmed cell death, and this ensures that damaged sperm aren't fertilizing an egg, which could pass down the damaged or dysfunctional DNA to a next generation. So I just thought it was interesting to include this to show it's regulating that way too. And then temperature also can affect the ability of sperm to mature and therefore fertility. So the ideal temperature for sperm to mature is at 34 degrees Celsius, and this is below body temperature, which is 37 degrees Celsius. So sperm maturation takes place in the testes, which is located in the scrotum, which is held outside of the body to keep the temperature cooler and able to develop sperm. There's situations where too much warmth of the scrotum can negatively impact the development and production of sperm. This could be if someone's wearing too tight of clothing, it could hold the scrotum too close to the body, which keeps them warm, or someone who uses a sauna a lot. And then there's also a disease state where one of or both of the testes fail to fall into the scrotum. So this will have a higher temperature for that testes, and it'll increase the risk of infertility depending on how many or which of the testes are not fallen. And then this is my worksheet for the student. I think it's important after watching lectures to quiz yourself on details to make sure you're understanding the concepts and how they're connecting to each other. So I think this would help after learning the material to kind of connect ideas and review. But thank you.